Yep, thank you so much for a nice introduction. Hi, everyone, and I hope you had a nice lunch and then great time so far in the conference. And then again, thank you very much for joining me today. Yeah, this is my first talk in PyCon and my second time in the PyCon Italy. It's so awesome. And today now I'm so excited to, I think it's better here, and to talk about how to build a machine learning platform in biology, which is open source. And yes. So let's start with a little bit about me. So my name is Furkan, and I'm from Turkey. My hometown is from the Cappadocia region. Maybe you see from some images. And I'm working as a researcher and data scientist in a biotech company in Munich. By training, I'm a molecular biologist and geneticist, so my background is full of genetics. And previously, during my bachelor, I worked as the computational biologist in a rare disease research lab, and then followed by a data scientist position in a biotech company, again in Munich. And now, yeah, that's me. So here is the agenda for my talk. We will start with some basics in the biology, because I thought it would be really nice to give some introduction, because that would be nice. So then everybody is on the same page. So then we will talk about the machine learning in biology and the applications and the problems over there. And then we will talk about the OmicLearn, which is our platform. And then I will present the demo, and then hopefully I will get your questions at the end. And let's start with the basics. Again, no stress. So the high SQL level of biology is more than enough for me. If you know what the DNA is, that's fine. So to explain better, so I present it in this way. So I make an analogy. So right now we are in the cell scale. Okay, this the gray circle represents the cell, which is the smallest biological unit. Okay, so you can think it's like a computing unit. And then we have our DNA, and the central dogma, it's like a phenomenon in molecular biology. So we have it dictates how the genetic information flows. So we have DNA. You can think the DNA is your .py source code. So if you collect all of your source code, we call it genome. And then this DNA is transcribed to RNA. And then you can think the RNA as an intermediate molecule, and it's like an interpreter. And then at the end, we translate the RNA to protein, okay? And then the protein makes the actual job. So then you can think this is the executable. I hope this analogy makes help you in a way. And to give you a sense how complicated things are, because from this image, it looks like a super simple, but I will give you one bonus point. So you know already the find and replace functionality in your favorite IDE is like a super, not something special, right? It doesn't worth it to talk about. But having the same functionality in our genome, so like a search and the replace in our genome ends up in a Nobel Prize in 2020. It's just 3 years ago. It's just crazy, difficult things. And now we are a bit zooming out, and then we were in the cell scale. Now we are talking about the samples. So I will go step by step how to translate from bases to bytes, like the computational way. And then we can work with several different sample types. So that could be the blood, or maybe you're already familiar with the saliva from the genetic test. And then the good point is that each sample has a different set of the steps for preparation. And then for the actual quantification, or the way of translating the biological sample to a data, this is how it becomes. And we are having several different machines. So I will not get into any details, because it's completely different fields, each of them. So we have a lot of the areas that are just focusing on them. And at the end, we are ending up at the biological data sets. And then we have this omics. So omics is a discipline working on those biological data sets. As you see, there are different layers. So genomics, epigenomics, transcriptomics. So they all refer to different part of the biology. So genomics works with the DNA and the gene. And right now I'm working in the field of proteomics. So, but the ultimate goal in the field is like interpret all the different layers of the omics data sets in a way that we can understand more the biology or the reason. And so I will also give some few milestones where we were, and then right now also where we are. And it starts like, I think the Pandora started with the Human Genome Project. It was early 1990, and it takes us like 13 years to complete the human genome. And it cost us like approximately $3 billion. And with the help of the thousands of researchers in 6 countries. And again, for showing the complexity, the last year, the full genome like released in the March 2020, and some gaps are filled, but still there are some gaps in our genome. So this is how complicated the problem is. So in terms of the cost for the genome, so in the 2001, it was just costing one human genome was $100 million. Right now it comes to the $600, but the aim is like ending up is $100 and democratize the human sequencing for several reasons. And for giving a better sense what data, like a volume of the data, I picked this report from the European Bioinformatics Institute. This is the latest report they released, and this is a consortium where a lot of researchers, research groups, academics, institutions submit their data. And in this graphic, you see the daily request per day, and how many requests is, like, the number of the requests is increasing dramatically. And keep in mind that this is only one consortium. And the number, in terms of the volume, they already, like, as you see, the increasing trend over the years. So every year, it's just like last year, they deposited 25 petabytes of data, and this includes images and some other data. So we talk about the data and how we translate from the biology to the computer. Then there are a lot of applications of the machine learning in the biology. So I think, familiar with the images from the COVID times. They were like scanning the tomography scans and apply it. And there are also some other fields in the text and audio. But today I will focus on the tabular data, which I'm currently working in. And the ultimate goal is like helping the clinicians at the clinic and then diagnose the disease earlier as much as we can. So at the end, we develop a platform, but first of all, what are we working with? So how does my data look like? So here, actually, the basic data that I have, like I work in daily manner. So we have the samples from the patients, and in this case, this is Alzheimer's data sets. And we have also the control groups, as usual, like the healthy groups. And in the columns, as features, we have the proteins, because I work in the field of proteomics. And for sure, you can have some more data from the clinical, so it could be smoking status or the sex of the patients. And for sure, you have the target to classify them. So this is the what section. So, and then there should be also the why, right? So why we develop this platform? So first of all, identify the patterns. So we would like to diagnose the disease, right? So this is a really crucial task. And one way to do is discovering a biomarker. So biomarker is a biological molecule. This gives a sign of this, the state of your health and disease status, okay? So one cool example is the BRCA gene. If a lady has a harmful mutation in those genes, their chance for getting a breast cancer and ovarian cancers dramatically increases. So the early diagnosis is saving the life. So we would like to discover biomarkers and for sure help the clinicians. And another point, again, the problem with the researchers during the machine learning applications is the misleading metrics. In this example, I just give it, but it's very common. If you have like such data set and such case, so 90 healthy and 10 patients, and the model like gives such a 90% accuracy, but the F1 score could be 0. But guess which metric is most reported in scientific papers? And the other one is, most importantly, is machine learning pitfalls. And there are 3 of them. The most important one, But one is the methods. So we are not able to, most of the time, the researchers are not submitting their codes make them on open source. So then we don't know what method they use, what hyperparameters they applied. So this is like a complete lack of transparency. And I've put one example from a research paper. As you see, there is no a way to reproduce this model. And the second reason is data leakages. So this is really crucial issue in the application of machine learning. And I found a really nice paper, review paper, recently published. So then they list a lot of papers, what the problem they have, and then they have the train test, they don't have train test split, or they do bad practices in machine learning. And the last but not least, some methods include stochasticity. So the randomness is another important point, but they don't define a certain environment that you can reproduce the results. So what's wrong? If you ask me what's wrong in the machine learning, in biology, there are a lot of problems, but I think the main problem is unreproducibility. So I believe reproducibility is the key in the science. And also another point is it should be reproducible for all people, not only the others. So I think you are familiar with it works on my machine. It's also the same for the researchers. So they said, okay, it's reproducible on my machine, then yeah, but we can't ship your machine, right? So far we talk about the what and why we develop this platform. Now let's talk about the how. I think this is the most important for Pythonistas. After listing the several reasons why we should develop this platform and the needs of the biologist, so first of all, the code should be open source, right? And the second is reproducibility, and also should be really easy to use by the biologist. Then we end up with OmicLearn. So then you can reach out on omiclearn.org. And yeah, here is my architecture. So in the OmicLearn, sorry, I will. In OmicLearn, we have the 6 components. So first of all, as I showed you in the what section, so we are working with tabular data sets because the output from the machines for the data sets are in CSV format or the Excel format. So obviously we use the NumPy and the pandas for analyzing and processing them. For machine learning part, obviously we use the scikit-learn, but we also included the XGBoost. So we search around for other methods, also even tested the deep learning for tabular data sets, but after our research, XGBoost was the most performant one. And for the web interface and the visualization, because the biologist should access the tool easily, we use the Streamlit. And then I will explain why we use Streamlit and also the Plotly and XGBoost in the following slides. And obviously, also, we use the Plotly visualization because the researchers want to get really paper-qualified papers. What they want is just download the figures and then put into their paper. No more work is needed for biologists. So, and then for documentation, we use the Sphinx. I will also show after my demo our documentation, and then we combine the Sphinx to read the docs. And for installation, sometimes we realize that biologists don't want to deal with the Python environments and some Conda environments. They just would like to test. But we deploy into the cloud, but there's also another problem. Sometimes biologists don't want to leave their data to cloud because they are just not willing to, and then they just would like to test the platform on their machine, because in that case, the data doesn't leave their machine. And then for this, we install, use the PyInstaller. Then we created the images for macOS and also the Ubuntu and also Windows. And then we also use the integration test. I use the PyTest for integration and the unit test. So because we have to be sure that once we update the packages, the results should be still reproducible. And then all the flows are going on the GitHub Actions. So that's the architecture behind the OmicLearn. And here I would like to also quickly show how we organize our files and the folders. You can for sure find all of them in omiclearn.org. The reason I'm showing that, maybe if you would like to build your own platform for your own field, so maybe you can just have a look. So this is the way we found out that works best, but we are open to any suggestions from you. And I think I talked about all the technologies in the previous slide, but there is one thing left. So the deployment to the clouds. Then previously we used the AWS, but then the Streamlit announced that they have their own cloud services. And then we sync at the GitHub repository on the master branch, and then it automatically catches the latest build from the master. So once we develop something and then put it into master, so it's automatically deployed, and then I can suggest you to do so. So far, we talk about the main architecture, but I would like to also talk about why we choose some specific tools, because those are some, after our experience, I thought it would be nice to give the sense. So for the Streamlit, actually, the main point why we're picking the tools is we check how much time we have, what is the human resources we have, and then what we would like to do. So for sure, we could also develop a web app platform using the Django or Flask with the React or Angular or the Vue.js, but we don't have that much human power, also that much time. So we need a rapid prototype. So for Streamlit, this is actually the tool you need. So as you see in the black, the code, so it's just 2 lines, and then we don't need any callback. So you can just make a dropdown, and then it's automatically assigned to the variable, and then you can collect all the variables, like the defined variables at the end at somewhere. And then there is no HTML, CSS, Python mix, it's just Python, so it's just so easy to use. And also, there you have a free deployment option. And another cool thing is that you can use the Streamlit with the other data stack, with like pandas, you can show your data frame, and then obviously we need also a lot of plots to show, and it also works very well with the Plotly. So this is the rapid prototype. It's just super easy, but everything comes with pros and cons, and there's also the cons. So for sure, it's not that much flexible, like as in like a native web solution. So you can't develop, like you don't expect to optimize everything, but this is the reason why we have the rapid prototyping. In terms of the performance, I think we use several other tools like a Plotly Dash and also the Bokeh and also some other platforms, but for sure it's not that performant as in the native solutions, but it is still good enough. Then I think you will realize how good it is, and then you can decide on your own. But the bad thing is the testing, the UI. I use the Selenium for testing the UI, but unfortunately I suffered a lot, and today I can recommend you to not use, and then because today my audience who attend me, I want you to live longer and have better eyesight. So yeah, don't try UI tests with that. And for the machine learning side, we use the XGBoost. The number one reason is our data, the biological data, is not a linear data, so it's just so complex. So then it has to capture the nonlinear data patterns. And also it should work with the sparse data, because there are several reasons, technological reasons, that your data could be null for those several values. But you have to, for sure you can impute, but at some times you need also such support. And it is fast, and then you have a chance to regularize your model in a way that they are not overfitting. Last but not least, why we use the Plotly, is that our researchers wants to just click the one button, download the graphs and charts, and put them into their paper. I will show them in example cases. So the reason is they would like to have the publication ready plots. And then for this, we need a lot of customization, and Plotly supports all of them. And then also another cool point is it works very well with the Streamlit. And then it also offers a lot of interactivity. So far we talk about the what, why, and how, and now it's about the highlights of our project. So once we finish up the project at last year, so our app was selected App of the Month by Streamlit, and it was a good success for us. And then last year in December, we published our paper. You can find it again on omiclearn.org. So, and it's open access, you can directly access, and in 6 months it got 4,000 views. I think it's a good number for a research paper. And then, last but not least, our paper is also featured on the cover of the journal. So that was super exciting for us. And this is the slide where I'm most proud of. So we have a lot of customers, which means our researchers. So they already used and cited our tools, and then they already discovered some new proteins, and they also check how they are correlating, so the machine learning results correlating with their normal classical statistical approaches. So we have a lot of reports, the research reports from a lot of fields, and then you see a lot of application sites. And as you see, they just directly download the graph from the platform and then use it, and then they are happy. And for sure, as a junior, it wouldn't be possible without without their help. I have to thank all of my managers and supervisors. And now I have the final presentation before the demo. So we recently released the last version in early May, and you can find the code, paper, and the platform and the documentations on omiclearn.org. So let me go over our tool. Can you see? I think it's visible. So in the tool, we have two sides. One, the left hand, and the right is the main window, I would say. So in the left-hand side, we have a lot of the custom optimization. So you can check different scalar methodologies and also check your feature selection, because I showed you in the what section, we have a lot of features. So it's not easy to train a model with that a lot of features. So you can also pick different classification methods and then pick as much as which one you want. And then the cool thing is that we had a nice documentation how to use those methods and how to go over what to click, what to not. And we have also the recommendations, because it's easy to overfit, and also we should show them how to interpret the results. So they are all reachable on our documentation. And once everything is set up by the biologists with just some clicks, and then, for instance, in this case, I'm using again the Alzheimer data sets, and then I'm trying to guess who is Alzheimer, who is not, based on some proteins, and then I just don't want to go into so much details, but I'm okay. I would be very happy if you have any questions, just ask in the Q&A. And then once you click the run analysis, so it makes all the modeling part and the training and then the validation. And this is actually the part where the researchers are looking for, because those are the candidates for biomarkers. So this plot shows which features contributed most to the model. So in this case, the first protein was, I know it's an ID, but once you just search for this protein ID in this biological database, so it is the tau protein, which is already well known for the Alzheimer's disease. So, and as I showed you, there are all other plots. And another cool thing is that what the researchers are very happy is our summary text, because I said the reproducibility is the key, and then they can just directly copy and paste the method section and then revert and then put their paper. And another cool thing is that I think if you remember in the Streamlit section of the code, I was saving all the variables into a dictionary. The reason is I would like to export them at the end, and this is called session history. So the researchers, we save all of options and the selections in the session history, so then they can download and then replicate and their results. So that was end of my presentation. And grazie mille and çok teşekkürler. So if you have also further questions, you can reach me out on Twitter, LinkedIn. I'm very happy to answer or help with your project. Yeah, thank you so much again.
Okay, thank you very much. Great talk.
Yep, thanks so much.
For me too, because I'm in the field, so very nice. There are several questions.
Okay.
So, some of them you kind of already answered, so I'll start maybe from the other ones. Okay, this is more of a philosophical question. That is, often happens in research that after a paper is published, the software is abandoned.
Yeah.
How do we deal with this? I think no one has the answer, but let's try.
Yeah, actually, this is also very common in the field of the bioinformatics. So the publications are done, and then few years later, so the project is gone. But this also was about to happen. Then we migrate our projects to academic institution, and I still contributed to the project, and then I'm keeping it up. And then I would be very happy if you cite us. So that give us motivation. So, but yeah, we are trying to do our best to support the tool and then make it like a live.
Great. Here's, well, you kind of answered, because the question is if OmicLearn have already been used to discover any important or well-acknowledged biomarker, but you show some research based on...
Yeah, exactly. So I can show you again. So there are some biomarkers are founded, and then they were also overlapping with the researchers already applied their own strategy. And in the second case, they put them asterisks. So it means that those asterisk proteins, the proteins marked with the asterisks, also overlapping with their research. So the tool performs and results in the good biomarkers, I would say.
Great. Okay, then we have a nice question that is, how much should biologists know about the technicalities or functionality of the tool to use it?
How much they need?
They need to know. So
yeah, I think that's a really good question. So therefore, we prepared a using page for this, and also the recommendation. We put actually a lot of efforts, because I believe it's not easy to use to directly apply your machine learning model to the data sets. And then there are lots of pitfalls and then the bottlenecks and during the application. So it's not that much high in if you would like to develop your own platform or write your own code. But in our platform, we try to make the user interface really easy to follow, and then we combined all of them in documentation and then explain them in basic terms and how you can navigate. We've also provided examples, and then we also linked the papers who use it. I think this would help researchers who would like to utilize our tool. I hope that answers the question.
I think so.
If it's not, just find me, and then I can elaborate.
A couple of more, and then we finished. Is there any functionality related to hyperparameter optimization? Yeah,
that's actually what we would like to do. So I want to use the Optuna, but the important point is that we are using the Streamlit clouds, and it's not that possible to use that much resources in that free, but we are planning to add actually some hyperparameter optimization so that the researchers find it, use it on their local machine, but not on the cloud, because there is the reserved place for us. Yeah. But it would be nice.
Okay. And finally, someone is asking, what is the revenue model?
Revenue model? So it's like an open source project, so we don't have any revenue model. So yeah, this is just for giving back to the scientific community. Yeah.
Okay, thank you. If anyone has any other question, okay, so thank you very much.
You're welcome, and thanks for joining.